problem in making tools is it's the old, you think if you build it they will come, but you have to go a lot further. You have to really get reagents into people's hands and really sort of make them easy to acquire. We've been able to come that far with the kinase sensing technology, with the protein-protein interaction technology. We have intellectual property on those reagents, but we need to get some more seminal demonstrations for people to say, you know, I can use that. But we also have to go a step further, because the average biology, biochemistry group will not go into the lab to make their own reagents. They want to be able to buy a kit that includes everything they need to do to use the reagent. So we've done that in one area, but not quite in the protein-protein interaction. So I think there's a path, but I think it has to be done, and that's how you have to get reagents into the hands of other researchers. For example, in nature there are many small protein modules. There are only 100 amino acids. They're little compact domains. They appear in a lot of more complicated proteins. Those domains, their job is actually to recognize small sections of peptide from other proteins. Sometimes they may recognize the carboxyl terminus of another protein, or a phosphorylation site on another protein, or a different kind of marker, a polyproline motif on a protein. So what we would do with our reagents is we would be able to make the segment that's recognized with a fluorophore in it that's going to be a reporter, and we could literally look for the interaction between that small peptide module with the fluorophore and the partnership and the partner domain. What that then means, if that partner domain professionally in the cell binds to something and triggers an activity, we'd have the short peptide there. Once it makes the physiologic interaction, you'd bounce the reporter off and you'd change a signal. So in a way, it's a way to diagnose interactions by having small reporting modules that will tell whether a domain is engaged or not engaged. And we have applied these tools in a very nice study in a neurologic system looking at interactions in the postsynaptic density, and really been able to prove that these tools are very, very useful for diagnosing physiologic interactions with minimal disruption. So, as I said, kinases, it's now realized that the aberrant or misregulated activity of kinases is a very, very common marker and source of disease. Because what happens when a kinase is overactive, it will be, let's say it's a traffic light within a pathway going to the nucleus. Kinase is sitting there. When it's green, the kinase is on. When it's red, the kinase stops, and no signal goes to the nucleus. If the traffic light is permanently green, the kinase is always on, and the signal to the nucleus is just going, and the nucleus is there to make proteins, to order the production of proteins, so cells just proliferate in an uncontrolled way. So you have a direct relationship between any kinase on that pathway to the nucleus and its activity, regulating and being in control. So if kinases are always on, there's a problem. So where inhibitors would be useful is they would dial down the activity of that kinase, basically turn the stoplight to red, or at least to yellow, to slow things down a bit. There are kinases that are known as receptor tyrosine kinases, which actually are planted in the cell surface, where they receive a signal from the outside, and then there are interactions on the intracellular domain of that membrane protein, which then cause a phosphorylation and triggering. So receptor tyrosine kinases are very much ones that there's a great focus on. For example, the epidermal growth factor receptor tyrosine kinase is a very, very important target. And people are really, the range of kinases now in the kinome is codified. We know the number, and there's so many relationships between each of them and disease, it's sort of mind-boggling. So people now need to be able to have tools to diagnose those activities. A lot of the tools that are available look not to whether a kinase is more active or less active, but to look to see whether it's in what's called an activated state. The trouble is those activated state markers are often surrogates for activity. They're not really activity measurements, but the technology that I've put in place actually measures activity. It really quantifies the active state. So I think that in modern biology, we need much more than a yes/no answer. Oh, it's activated, so it's bad, and it's not activated, so it's good. We need metrics, we need numbers, we need relative activities in real quantifiable ways. And that's what fluorescent tools can do for you. They can literally give you, oh, this is 80% of activity, or 70 or 60, not just looks active, looks not active.